Hi, everyone. Welcome to the last lecture before the second midterm. We're going to complete our discussion of the sensory systems of the human and animal nervous system today by talking about the somatosensory nervous system that allows us to feel and detect the location of our body in space. And we'll also talk about the intricate link between all these sensory systems and our motor output system, which we've talked a little bit about. And so we'll talk about the sensory motor connection a lot throughout the lecture today. So we'll talk about 5 topics. First, the idea of movement control as a hierarchy where information is sent from high levels down to low levels where there's muscles to control them, kind of like a puppet master. We'll talk about the organization of the motor system. We'll talk specifically about the basal ganglia and the cerebellum and their role in the regulation of our motor output. And then we'll talk about the somatosensory system and the remaining 2 topics. We'll talk about the types of receptors on the skin and the pathways the information takes to get up into our brain. And then we'll talk about the somatosensory cortex behind the motor strip and how it processes this touch information. So our movement control is governed by this hierarchical system that involves the cerebrum, our brain, which involves our conscious decisions to say, get out of bed and go eat breakfast. And this information is sent down through lower and lower level parts of our nervous system through the brain stem down to the spinal cord, where it's then sent out to our muscles to produce movement. When we get damage to the brain stem or spinal cord, we can still imagine the movements we want to make, but we can't produce them. The output structures along the way are damaged. We can think about the sensory motor process as following these roughly 8 steps. We see information with our eyes that we want to reach out and grab. Our brain might make a plan for how we're going to reach out and grab that object. It sends that information down through the spinal cord to the hands, and the muscles of the arm and hands change to move the hand and grab the cup. Then sensory receptors on the fingers and in the joints of the hand detect that the cup is in the hand and maybe the temperature of the cup as well, and transmit that information up into the spinal cord. And then that gets carried up into the brain. Along the way, the basal ganglia and the cerebellum can act like judges or moderators of this force. So if they detect too much or too little squeezing, they can adjust this downward control of the movement. And then finally, the sensory cortex receives the information that the cup has been grasped. So we see this loop between our perception and our action, and then our perception of the feedback from that action. And in addition to that loop, there's this basal ganglia and cerebellum processes happening to modify our movement along the way. So we'll talk today about the basal ganglia, a subcortical region that is involved in deciding the appropriate amount of force to use in different situations. And we'll talk about the cerebellum, which we'll see regulates the timing and accuracy of our movements and can help us learn on the fly. As we've learned, afferent information is being admitted into the nervous system. So the somatosensory information is afferent, and it travels up into the nervous system. And movement information in the opposite way travels down as efferent down to the muscles of the body. So we have incoming sensory information, say here when you step on a tack, up to the brain, and a signal down to move the body. We'll talk a little bit today about how the output that we make with our body is parallelized in that we can output multiple motor plans at the same time. I can talk, and then at the same time, I can move my hands and adjust the slides. So these aren't serial processes that need to happen one after the other. So this incoming sensory information enters the central nervous system via these axon fibers called dorsal root fibers, which we'll see. They're in the posterior part of the spine. Fibers leaving the anterior part of the spine, closer to the abdomen, carry information from the spine out to the muscles. They also bundle together into what's called a root. So there's an anterior and a posterior root, as we'll see, coming out of each segment of the spinal cord. So here we can see the fibers entering the posterior side, bringing information from the touch receptors of the skin and joints. And we see the motor information being sent out on the anterior route to the muscles. You can see that the sensory information can ascend up to the brain, and it can also cross over to the contralateral side of the spinal cord, where it can influence the motor neurons there and also ascend to the brain on that side. And we see that these white matter tracks, so this is a segment of the spinal cord as we learned in our chapter 2, and the white area is the axons and the gray area are the cell bodies. These white axons represent highways up and down the spinal cord, sending information to and from the brain. The cell bodies themselves are only in these dark gray areas that form a butterfly shape inside the spinal cord. Much like our ancient relatives, the worms and snakes, our body is segmented, and each of the segments of our body is innervated by one of these nerves coming in and out of our spinal cord segments. You can picture humans walking on all fours to better understand how these dermatomes, they're called, these sections of the human skin that all send their incoming sensory information into one root of the spinal cord. You can see how they're distributed on the body. And so each spinal segment, each axon entering the back of the spinal cord brings information from a different section of the skin. And each spinal cord segment sends information out to muscles in a particular part of the body. And the body surface corresponding to one spinal cord segment we call a dermatome. And so when you get damage to one of these spinal cord segments, you often see specific pain or discomfort in one of these dermatomes, or constrained inside these dermatomes. As we've seen, if we look through slices of the motor and sensory cortex, we see differences in the morphology of the cells as well, relating to their function. We see that layers 4 and 5 are different in the two types of cortex. In the motor cortex, or let's start with in the motor cortex, the output layer, layer 5, is relatively thick compared to in the sensory cortex, whereas in the sensory cortex, this input layer, layer 4, is relatively thick compared to the motor cortex. And this makes sense because of the increased amount of incoming information to the sensory cortex and the large amount of outgoing information coming from the motor cortex. We've seen in chapter 2 and in your past classes that the motor cortex is just anterior to this central fissure, and the sensory cortex is just posterior to that central fissure. Because of how quickly we can perform skilled movements, you can think about the hockey game last night, the amount of movement you need to ride a bike or to run across the street, call out to someone while you're doing it. We can't possibly wait for each movement to complete and see how it goes before we start the next one. This is much too long of a time to sequentially sequence these actions together. I'm going to reach out here, and then I'm going to turn my hand, and then I'm going to punch my fingers together. There must be some other mechanism by which motor plans are chunked together to effectively control our body in this way. So movements usually are performed not as single movements and then waiting for feedback, but as a sequence of movements that are grouped together in order. And the next sequence is held in readiness online, waiting while the previous one is going on. So a motor sequence you can think of as a movement module, a set of movements that the brain is pre-programmed to make as a unit. So most of our learning to read or write or ride a bike or to play piano, is a sequence of actions that we need to learn to string together successfully. As one sequence is being executed, we need to get ready for the next one to be occurring so that it's completely ready and can say the next sentence that I'm going to say can come out smoothly after the first one is complete. Language is a very good example for this sequencing of motor outputs. So we have the prefrontal cortex involved in planning the behavior at the highest levels, the premotor cortex, which sequences together the specific amount of movements that are going to be needed. And then finally, the primary motor cortex along the way, which specifies how each movement is going to be made, which particular muscles and which groups are going to be needed to produce the sequences and the plan specified by the higher levels. So we see the prefrontal cortex planning movements, the premotor cortex organizing them together into sequences needed to carry out those plans, and then finally the motor cortex executing them with specific actions. Notice here the prefrontal cortex is this giant section of the brain, and we're saying that the whole thing plans movements. Generally in the section of the course after the midterm, we're going to cover more of the roles of this frontal lobe and our cognition. And we'll see that it's not just in planning movements, but in lots of high-level cognitive behaviors that are uniquely human. However, when even animals that have damage to their prefrontal cortex show very high-level disorders, in this case, they can have an inability to sequence particular movements together appropriately. So here in this task, a healthy animal learns to poke food through a hole and catch it with the other hand underneath. But after 5 months with a lesion and trying to learn the task, this other monkey without a prefrontal cortex that's functioning correctly can't string together the two movements in order to get the reward. Our primary motor cortex is specialized in producing very skilled movements like writing with a pencil or typing on a keyboard. And if you have damage to this area, you have damage reaching and grabbing things correctly, shaping your fingers to the correct objects, and making correct hand grasps in the right situations. Here are two examples of different stereotypical human grasping types. So there's evidence that of this hierarchical nature of our motor output, that is, that the prefrontal cortex commands lower levels to execute movements. So the prefrontal cortex can formulate a plan of action, and it instructs the cortex to appropriately sequence the behaviors together, and then the primary motor cortex, as we say, can execute these movements together. And there's evidence from fMRI studies as well of this hierarchical nature. If you just ask participants to make a particular finger press with one of their fingers, you'll get a slight increase in blood flow in the motor and sensory cortices associated with that finger. If you ask the individual to perform a sequence of movements with that finger, then the premotor cortex here comes online and is involved as well, because of this need to sequence the movements appropriately. And finally, If you ask the participant to have this very high level goal of finding a route through a maze, but make the similar low level behaviors with their finger, additional areas in the prefrontal cortex and the temporal lobe are activated, indicating this complex goal-directed behavior recruits more brain regions. As we move down the hierarchy, we can see that the brainstem has a particular set of neurons that are involved in the production of species-typical movements. Species-typical behaviors are behaviors that are performed by all members of a species, like the pose that a cat makes when a dog runs up to it, or the behavior that a chicken makes when it gets scared in the coop, or a rooster in the morning. So stimulating different areas within the brainstem can produce slightly different species-specific behaviors like these. Some of them involving turning the head or walking or running, and lots of them involving aggression or fear as well. The stimulation of these regions and the effect on the subsequent behavior also depends on what type of external objects are present in the environment, indicating these tight connections between the visual cortex and these areas in the brainstem. If there's a particular stimulus coming into the visual cortex, these brainstem regions will not produce the same amount of behavior given the same amount of stimulation. You can see that the duration of stimulation also influences behavior, that if you stimulate the brainstem of this chicken enough, at first it will just be produce threat behavior, but then if enough, it will just fly away altogether. So the brainstem seems to be producing these complex behaviors that are adaptive for our species survival, including maintaining our posture, standing upright, coordinating the movement of our limbs when we swim and when we walk, and we groom, get out of the shower and dry off, and when we make our beds. As we continue to move down the movement hierarchy, we'll talk about the spinal cord, where this information is being sent after the brainstem out to the muscles of our body. Christopher Reeve was an actor who played Superman in a number of Superman movies that were filmed in Alberta. He fell off a horse and severed his spinal cord near C1 and C2 in the neck. This left his brain functioning and his spinal cord functioning, but they no longer were connected together. So he became completely paralyzed. And he couldn't even breathe without assistance because the signals being sent to breathe are being sent through that part of the spinal cord from the brainstem to the lungs. Quadriplegia is what Christopher Reeve suffered from, as a paralysis and loss of sensation in the legs and arms due to spinal cord injury. A paraplegia is a paralysis in the legs and lower body due to a spinal cord injury that occurs lower in the spinal cord. Interestingly, the spinal cord below the lesion can still function even without being attached to the brain. So spinal reflexes can still occur. And so the paralyzed limbs can display spontaneous movements, but the brain can't control the timing of these movements at all. And these spinal cord functions are enough that you can even get an animal or a human to walk on a treadmill based off the walking reflexes present in their spinal cord, which was something that Christopher Reeve was able to do, if their body weight is held up. Okay, next we're going to talk a little bit about the organization of the motor system and how activity of the motor system can produce the wide array of different movements that we could make. over 100 years ago, it was discovered that electrical stimulation of the brain of dogs that were anesthetized could produce movement of their body in different places, and usually was found on the opposite side of the dog's body, in the contralateral side. Almost 100 years ago, Wilder Penfield in Montreal continued this idea by electrically stimulating humans who were undergoing neurosurgery, and using the subsequent reports from the humans during their waking surgery to map out the cortices of their patients. And this research confirmed the role of the primary motor cortex in producing movements. By stimulating different areas, Penfield was able to show different activation of different parts of the body. And out of this research came the idea of a homunculus, this little representation of the human body distributed in our motor cortex. And this relates back to this idea, we've talked a little bit about it in the class, about retinotopic and tonotopic organization of our sensory systems. This is another similar representation of a topographic organization. There's a spatial representation of the body in the brain. And one interesting feature of the homunculus, which you'll remember having seen the picture, is that the hand, feet, lips, and tongue are disproportionately larger. That is, the area of cortex dedicated to processing and making those parts of the body move is larger than the parts of the brain dedicated to making, say, our chest or our abdomen or our back move. So the motor homunculus represents this map of the cortex, map of the body inside the cortex. And because of how fine scaled and how many sensory receptors there are in our skin and our face, they have a much larger representation in the cortex. And vice versa, things like our trunk and our neck, which have much more coarse representation, have a smaller representation in the homunculus. So the distortions associated with the homunculus illustrate that how innervated these areas of the hands and feet and lips are. And so therefore how much real estate all those axons coming from all those neurons needs in the brain to receive that information. The early idea was that the homunculus was like a puppet master. That is, that you could think of it like Geppetto controlling Pinocchio, and that the frontal lobe could control each of the various muscles of the body when needed. And so this was a simple idea that when the cortex wanted to move the arm, it would activate the arm area and the muscles of the arm would move. However, recently it's been shown that the motor cortex isn't necessarily representing just single muscles that can move, but rather a repertoire of behavior, much like the activity when we stimulate the brainstem elicits these species typical behaviors. It looks like the primary motor cortex seems to contain patterns of learned behaviors. And so stimulating the premotor and the motor cortex of a monkey elicits these complex sets of movements like climbing or descending a tree, reaching and grabbing, defensive postures, and bringing the hand towards the mouth. It's generally shown that more posterior stimulation in the primary motor cortex generates less complex behaviors, like licking or masticating, grabbing something with the hand, or grabbing something with the hand far away. So you can think of maybe these purple movements as being a couple of the blue movements combined together hierarchically. In the human MRI studies, it's shown that the motor cortex also seems to be organized into functional movement categories, so that different types of movement seem to be represented in different areas and that they can change with learning and practice. So the motor cortex seems to not just be a precise map of which muscles to move at which times, but more like a lexicon or a dictionary of different movements that we can make together, the waving of a hand or the different speech sounds. So the motor cortex of an animal at any given time can represent the entire repertoire of movements that it can make. If we look at motor cortex neurons and we record their activity more closely, we see that their activity seems to indicate that they're involved in both the planning of movements prior to their occurrence and the initiating of the movement start and the continued production of the movement. And finally, they seem to code for things like the force of the movement. So we'll see that they discharge before the neuron starts to fire before the movement even occurs. And when there's more force needed from the movement, the neurons fire more. And they also seem to code for the relative balance between our muscle groups. You'll recall that, say, the bicep and tricep form a muscle team called a flexor and an extensor, and they can code for the balance between these two as well. So here is the recording from the motor cortex of a monkey who's being taught to rotate its wrist to lift a weight up to receive a reward. Notice that the neural activity increases before the monkey starts moving, indicating some role in planning, but it also continues throughout the motor execution. When a weight is added to make it heavier and harder to move the wrist, an increased firing rate occurs, indicating that the force of the movement is also coded by the firing rate of these neurons. So motor cortex seems to be involved in planning and executing movement and also adjusting the force and duration. In human participants, some studies have shown motor cortex neurons active when no movement is occurring. So we can have kind of sub-threshold activation of our motor cortex. If you close your eyes and imagine hitting a golf club, you'll imagine that you might activate the same motor cortex circuits involved when you actually hit the golf club. And that sub-threshold activation of those circuits is an important feature of the brain, and it could possibly provide a way that we could create brain-computer interfaces. So if I imagine moving my hand to the right or imagine moving my hand to the left, I could sense that brain signature of that imagined hand movement using some brain imaging technology, and then translate that intended movement into maybe the movement of a cursor on a screen or the steering of a car. The motor cortex is incredibly plastic in that it changes with our experience. When we practice a skill, learn piano, our motor cortex can change. When it gets damaged, the motor cortex can try to make up for that damage. So a study was done in monkeys in which they found the hand area of the motor cortex and then lesioned a small part of that hand area, and found that without any rehabilitation, without a behavioral intervention making the monkey move around, the monkey lost their ability to move the hand. When they made the monkey move its hand around, and in addition to losing its ability to move its hand, they found that the motor cortex hand area became smaller and the adjacent elbow and shoulder areas became larger. We'll see a picture on the next slide. With rehabilitation, the hand area did not shrink and the monkey retained some ability to move the hand. So continued use of the hand, even with a lesion here and no live neurons in that area, continued use of the hand retained a bunch of the neurons that were involved in processing and moving those hands. So you make a small lesion with electrical current, and you find 3 months later with no rehabilitation that the hand and digit areas are much smaller. But with rehabilitation, they retain their size. And the way that this retention likely occurs is that existing connections between, say, the hand area and the elbow and shoulder area get to be utilized in this new brain as it learns to adapt. As we move down the spinal cord, we'll think about two pathways that information takes to get from the brain to the muscles. These are called corticospinal tracts, tracks from the cortex to the spine. These are the main exiting pathways that the motor information takes to the spinal cord. The axons descend to the brainstem. Some branch off and attach to different brainstem nuclei, and eventually they all merge on the surface of the brainstem and create these large pyramidal bumps. So they're called pyramidal tracts. There's two major tracts that we'll talk about today, sending information down from the brain to the muscles: the lateral corticospinal tract and the anterior corticospinal tract. These have different functions in that one seems to make the distal parts of our legs and arms move, whereas one is processing the movement of our core, our trunk. So the lateral corticospinal tract carries information to the muscles that control our fingers and arms, and generally on the opposite side of the body. So information will leave this motor cortex, go down the lateral corticospinal tract, and get sent out to this arm to move this muscle. The anterior corticospinal tract generally remains ipsilateral on the same side of the head, and it moves muscles about generally related to the midline, the pecs and abs and back muscles. So here's these two tracts in a nice diagram showing the motor cortex here with axons leaving layer 5, sending their axons down through the brainstem into the spinal cord. You can see inside the brainstem that half of these axons dissecate over to the contralateral side, where they descend, and we call this the lateral corticospinal tract, and it's contralaterally organized so that the right motor cortex sends information to the left finger to move. However, the right motor cortex also sends information to the right abdomen, and generally information in this anterior corticospinal tract synapses bilaterally, so it controls both sides of our core, making it very hard to flex one half of your abs. So here's a nice diagram showing the lateral corticospinal tract coming down from the contralateral hemisphere. It's already crossed over, and now the axons are synapsing on both these interneurons and motor neurons in the anterior horn of the spinal cord. You can see a small picture of the arm here with the hand, forearm, bicep, and shoulder, indicating that there's some anatomical topographic organization here, and that as we move down this horn, we go from the shoulder to the elbow to the wrist to the fingers. So different parts of the spinal cord send information to different parts of the body in a topographic way. Here we can see the anterior corticospinal tract, which came down ipsilaterally, synapsing generally on the midline parts of the spinal cord, which activate muscles in the trunk and the back. So you can see zoomed in this kind of mini homunculus inside the spinal cord. And so these information coming down from the axons of our motor cortex synapses onto these interneurons and onto the neurons themselves, and they send their axons out to the muscles to activate them. So these tracks originate in the neocortex and end of the spinal cord. They synapse onto interneurons and motor neurons. And the motor neurons finally carry all the commands out to the muscles. Inside the ventral horns are both these interneurons, which project onto motor neurons, and the motor neurons themselves, which project onto the muscles. We saw how the more lateral parts of the spinal cord process muscles that are more distal from the body and the fingers and hands. And the most medially located motor neurons on the spinal cord process or send information to muscles that control the center of the body, the medial parts of the body. So neurons in the motor homunculus in the left hemisphere control the trunk on both sides of the body and the limbs on the right side. As we talked about, our muscles come in pairs. The extensor muscle moves the limb away from the trunk, and the flexor pulls it in towards the trunk. And connections between the interneurons and motor neurons ensure that when our bicep contracts, for instance, our tricep is never also trying to contract. They inhibit one another. So here the tricep extends away from the body, the bicep pulls towards the body. They both release both have acetylcholine released on them by motor neurons from the spinal cord. Both project from different parts of the spinal cord. So the flexor and extensor axons are both projecting from the same section of the spinal cord. But there's also interneurons in here that inhibit one another. Okay, next we're going to talk about this kind of off to the side part of the motor system, these two structures called the basal ganglia and the cerebellum. Without these structures, all of those processes that I just mentioned can still take place, but they take place in this wildly unregulated environment where behaviors can go off in random ways or be stuck from occurring. The basal ganglia and the cerebellum adapt the behavior to be just right for the situation. The basal ganglia receives input from the whole neocortex, and it also receives dopaminergic input, dopamine input from the striatum and the substantia nigra. And it projects back to the motor cortex from here to send information out to the body to move. And the basal ganglia is involved in a wide range of functions, including learning of habits, motivation, emotion, and what we're talking about today, the control of our motor output. We'll talk a little bit about its role in habit learning after the next midterm when we talk about drugs. Here's some an anatomical refresh of the basal ganglia. You'll recall it's this headphone type structure with the part that would wrap around your ear being called the caudate. Caudate means tail. So just calling it the tail of caudate is kind of a funny, like saying chai tea. And you can see here in the cross section and a coronal section through the brain, that caudate here, and then you can see the putamen and the globus pallidus, kind of the sections that would just be on the outside of your ear, one on top of the other. We'll see that the globus pallidus is what ultimately inhibits or disinhibits our behavior based off its connections to the putamen and the caudate. We see that the substantia nigra, this incoming dopamine information, regulates these circuits as well. So the caudate nucleus and the putamen together form the basal ganglia, form the striatum, which is called striped body because of all these striped fibers running between them. The subthalamic nucleus is included in the basal ganglia, as is the globus pallidus. And there's these loops between the brain and the basal ganglia and back to the motor cortex, which are likely involved in selecting and producing the perfect movement for a given situation, and also likely given the correct emotional expression for a given situation, although we won't talk about that today. When you get damage to your basal ganglia, you can get two major types of syndromes. You can get a hyperkinetic symptoms, that is, you move too much, and you can get hypokinetic symptoms, where you move too little. This is a clue about the function of the basal ganglia. So if you get damage to the caudate and putamen, you can have unwanted movements and twitching. These are seen in things like Huntington's and also in Tourette syndrome. When you get damage to the basal ganglia in a different area due to degradation of dopaminergic input, you can get a decrease in motor ability, leading to rigidity and difficulty initiating and producing movements, like in Parkinson's disease. So one proposal of what the basal ganglia does is it acts like a volume or a gain function on our movement. A section of the basal ganglia called the globus pallidus internal acts like that volume control. If it's turned up, then movement is blocked. If it's turned down, then movement is allowed. And so depending on how active this globus pallidus is, movement will be either allowed or blocked. And the activity of the globus pallidus is affected by these two pathways called the direct and indirect pathway. The direct pathway inhibits the globus pallidus internal, freeing up the basal ganglia to produce more movements. The indirect pathway activates the globus pallidus internal and inhibits the production of movement. So here's a complicated looking graph that shows these relationships. So here's the normal connections between the cortex to the thalamus, and then back to the cortex and out to the brainstem. The globus pallidus here has this red inhibitory connection to the thalamus, so that if it's too active, it inhibits movement being processed through the thalamus out to the brainstem. And so the more active this area is, the more movement is clamped. Alternatively, when this is decreased, this inhibition goes down, and this is free to be active and process this information. So the more active this area is, the less movement there will be. And so you can see that the activity of the globus pallidus is this balance between the indirect and direct pathways. And the indirect pathway receives information from the dopaminergic system as well, leading to this imbalance in Parkinson's when the dopaminergic system breaks down. And in Huntington's, there's damage in this pathway leading to the opposite effect. The nucleus accumbens is an area in the brain that receives projections from dopamine cells in the brainstem, an area called the ventral tegmental area. And this pathway from the ventral tegmental area to the nucleus accumbens and then up to the frontal cortex is a second dopamine pathway, which we'll talk about next lecture about when we talk about drugs and reward, because this pathway is part of a loop that involves our perception of reward cues. So one pathway starts in the substantia nigra and goes up to the basal ganglia. And this other pathway goes up to the frontal lobes through the ventral tegmental area and the nucleus accumbens. These are two dopamine pathways in the human brain. What about the cerebellum? The cerebellum, and just one last final say about dopamine is that these dopaminergic circuits that innervate our frontal lobes can also have major influences on our movement. As you know, if you've seen someone suffering from methamphetamine addiction downtown, One of the telltale signs is just very erratic, very hyperkinesis. That is, lots of movements coming out without very much clamping. Okay, the cerebellum is another module of our motor system that is involved in modifying the motor activity in real time. The cerebellum has two hemispheres, left and right. It also has two sections, and it itself has a homuncular organization. There's a small map of the body represented inside the cerebellum. The more lateral parts of our cerebellum control movement of our limbs, hands, and feet, and the fingers, and the medial parts control the face and midline of our body. Here's a diagram showing that both the floccular lobe, which is involved in our balance and eye movements, and then also this major part of the cerebellum, the two hemispheres, and these two little homunculi distributed over the cerebral cortex, cerebellar cortex. Cerebellar surface is a better way to say it. So the medial part controls the face and midline, the more lateral parts control the distal appendages. And the pathways from the cerebellum project into the brainstem and connect to the downward going spinal cord connections that are going out to our body. So the cerebellum has two functions living in this part of the loop. So information gets sent down from our body to move, and then our cerebellum gets a copy of that command. And then when we move, we see and feel how the movement went, and the cerebellum also gets a copy of that feedback. And the cerebellum can take the intended movement and the feedback and compare them and see how close we were. And if we were off by a little bit, it can adjust the movement in real time. So the two main motor functions of the cerebellum seem to be related to timing. The cerebellum can precisely control the timing of our movements. And when we have a breakdown, we lose our ability to do this. And also maintaining the accuracy. So when we start to make errors in our movements over time, the cerebellum seems to be involved in maintaining their accuracy and making adjustments. One of the best evidence for this comes from a study of people with cerebellar damage who were compared to control participants in a task that involved throwing darts at a dartboard. Both controls and regular participants originally were just throwing the darts at the dartboard, and they were measuring the distance from the target. And they show, well, patients with cerebellar damage might have been a little more variable, but they roughly were aiming towards the center of the target. Then they put on prism glasses on these patients that make the whole world appear like it's shifted over to the side. And when the participant throws the dart, because of that prism shift, it goes way off where they intended to throw it. A control participant can see that and feel that discrepancy and slowly adjust it over time, such that over a number of trials, they are overcoming those prisms. However, when the prism glasses are taken off, that compensation is now an overcompensation, and they have to unlearn it as well. But the cerebellum seems to allow them to learn to adapt and then to learn to stop adapting when needed. Patients without cerebellums, however, with damage to their cerebellum, showed no improvement once they had the original decrement from the prism glasses. And then when the prism glasses were taken off, they showed no rebound effect. So the cerebellum was not adjusting their movement on the fly. So the cortex sends information to the spinal cord. A copy is sent to the cerebellum. The sensory receptors feel what actually happened and report it to the cerebellum as well. So the cerebellum has both what you wanted to do and what happened, and it can calculate the error and it can correct the movement. So the cortex sends information down to move the body, but it also sends information via the inferior olive to the cerebellum. The cerebellum receives that feedback from the body and can compare those two to create an error signal to adjust the cortex to improve on the next trial or the next throw. Okay, next we're going to move on to the somatosensory system. Now that we've talked about the motor system and how it can output and modify our movement, we'll complete our tour of the sensory systems of the brain, talking about the somatosensory system and also the vestibular system before ending by discussion of the somatosensory cortex. So our somatosensory system is involved in telling us what our body is doing, where it is, what it's touching. It measures touch, temperature, pain, the position of our body in space, and the movement of our joints. It allows us to distinguish between what the world is doing to us and what we're doing to the world. And because of the intricate link to our body, it has a much closer relationship to our movement than our other senses do. And without incoming sensory information from our somatosensory system, our movement almost breaks down entirely. The somatosensory receptors is the primary sensory receptor of the somatosensory system. There's areas of our body with larger numbers of receptors, and these are more sensitive to stimulation, just like in the visual or auditory domain. And so the different types of somatosensory stimuli are sensed by different types of receptors based off their physiology. Humans generally have two types of skin. We have the hairy skin of our forearms, and then this glabrous skin of our palms and lips. The glabrous skin has no hair follicles and it has much larger density of sensory receptors, which then get passed on to these larger areas of the homuncular sensory regions. Generally, we can sense these three classes of things. We can sense nociception, pain, temperature, and itchiness. These are generally done by free nerve endings, just open nerves that get activated when they move around by the chemicals in the extracellular space. Our body also has a sense of touch called hapsis. We perceive fine touch and pressure, and this allows us to, for instance, identify objects just by touching and grasping them. Generally, these work by mechanical stimulation of a hair or by pushing on the tissue, actually mechanically moves some receptor gates and opens them up. Proprioception is the perception of the location and movement of our body parts. We have stretch receptors in our muscles and tendons, and we have receptors in our joints that detect their angle as well. So much like a robot has a sensor for every one of its arms and which position they're in, our body has this as well. This chart shows those three major categories: nociception, hapsis, and proprioception. And it shows different types of receptors. These are the primary somatosensory receptors and where they are in the body and the skin. And it also talks in this column about their adaptation. Slow adaptation means that they generally detect long-term changes over time. Rapid adaptation means they detect quick, fast changes like tickles and pokes. There's a mistake in this graph in the textbook, and that this red block, Ruffini corpuscle, is actually a rapidly adapting. And I'll say in a couple slides from now where the text in the textbook gets it wrong as well. Okay, so there's free nerve endings for pain and temperature. We got all these different types of receptors and corpuscles to detect hapsis, and then muscle spindles, Golgi tendon organs, and joint receptors to detect the position of our body. So our somatosensory receptors tell us when something is happening. They tell us where on the body it's happening, and they also tell us whether it's still occurring. So the rapidly adapting receptors, they respond briefly at the beginning and end of a stimulus, and they'll stop being active once the stimulus continues to be present. The slowly adapting receptors, however, continue to fire as long as the stimulus is impinging on the body. Rapidly adapting receptors, like haptic receptors that respond to touch, are called Meissner's corpuscles, and fluttering sensations are called Pacinian corpuscles, and vibrations are also called Pacinian corpuscles. The textbook incorrectly writes Ruffini corpuscles here as something responding to rapidly adapting vibrations when they're not, because Ruffini corpuscles actually respond to slow adapting indentations. Other slow adapting receptors are like the nocioreceptors that respond to pain, heat, and cold, and the hair receptors. So you can think of this kind of rapid versus slow adapting receptor similar to the idea of differences between, say, rods and cones, or different frequencies along the basilar membrane. These are different features of the stimulus that the nervous system is designed to detect. So when we look at these sensory nerves, they have these long kind of extensions at the end and they're dendrites, but they have then an axon attached to them that goes into the spinal cord. And the dendrite and axon are continuous, and they carry the information from the skin into the central nervous system. So the very tip is what's responsive to the sensory stimulation. And each of the segments along our spinal cord has one large root ganglion where all of those posterior axons come in from all of the skin in that dermatome. And once they're inside the spinal cord, they can either synapse onto other neurons, or they can continue up to the brain to bring that information to the brain. And we'll see that here. So here's the posterior root neurons that have some sort of receptors on their dendrites, and they pick up some touch information, say, and they pull that information down their axon. It goes by this root ganglion where their cell body is, but it continues and goes right inside the spinal cord, where it either synapses onto a neuron there, or it ascends the spinal cord up to the thalamus to bring information to the somatosensory cortex. The neurons that carry fine touch have these large myelinated axons, and generally they ascend ipsilaterally up the spinal cord before they cross over at the thalamus and go to the contralateral somatosensory cortex. We'll see that's different from the axons that carry pain information, which instead cross over and then go up. So the proprioceptive and haptic neurons carry information about the location and movement and touch and pressure. And they carry this on large, well-myelinated axons, which means that the information travels fast. Opposite to this, the nocioceptive sensory neurons bring pain, temperature, and itch information along small axons with low myelination and therefore are slower. When you get local anesthetics, like when you've got dental surgery, it blocks pain through both pain perception by reducing activity in those incoming sensory nerves, but also that reduction of sensory information makes our face feel paralyzed. This process is called deafferentation. So loss of any incoming sensory input can lead to afferent issues with that structure. So if we lose our ability to feel in our hand, we'll have a decreased motor function in our hand. So there's long-term evidence that movement abnormalities can occur when we have damage to the incoming proprioceptive and somatosensory information. So in this case, Oliver Sacks describes someone who couldn't have any proprioceptive information about where their body was and had to constantly look where their body was. So they describe proprioception as being like the eyes of the body. And so then therefore they felt like their body was blind. As I mentioned, the haptic and proprioceptive axons that bring information into the spinal cord on one side ascend the spinal cord ipsilaterally on the same side, whereas the nocioceptive information about pain or temperature synapse onto neurons on that side, and then those neurons desiccate, they cross over to the contralateral side before they ascend up to the brain. So my left finger, if I tickle it, that information is going to go up ipsilaterally to my thalamus. If I hold a lighter and burn it, that information is going to connect into my spinal cord, cross over on the next neuron, and then go contralaterally up to my thalamus. And they're going to happen at different speeds. So here's another diagram showing that. Here's the incoming pain and touch information, and pain is in the dashed line. So whereas we saw the touch information comes in and it immediately goes up to the brainstem where it synapses and then crosses over in an area called the medial lemniscus, right here, medial lemniscus. And this medial lemniscus gets information from both pathways, right? So those pathways have joined back together. The temperature information or the pain information synapses onto a neuron whose axon crosses the spinal cord and then ascends into that medial lemniscus. So this axon, the axons of the medial lemniscus, their cell bodies are either in the contralateral spinal cord for pain information or in the contralateral brainstem for touch information. In both cases, the thalamus receives this information in an area called the ventrolateral thalamus, VLT, and this relays the sensory information up to the primary somatosensory cortex. Here's the primary somatosensory cortex, which is actually composed of 3 different Brodmann areas that look different under a microscope because of different morphology. Okay, so pressure and touch doesn't cross over, immediately ascends, synapses, and then it joins, as it crosses over, joins the medial lemniscus up to the thalamus. Nocioceptive information like heat synapses onto a neuron ipsilaterally, that axon crosses over and then ascends into the medial lemniscus before synapsing on the thalamus. So the pathway that immediately ascends, we call the posterior spinal thalamic tract. This carries the haptic and proprioceptive information. As we say, they enter and ascend ipsilaterally, and where they synapse is an area called the dorsal column nuclei. So right here is the dorsal column nuclei before they cross over and join the medial lemniscus. And then these axons from the dorsal column nuclei cross over to the opposite side and they form the medial lemniscus. And they synapse onto neurons in the ventrolateral nucleus of the thalamus, which projects to the somatosensory cortex. This is the same image. I'm going to now talk about the other route. So the route that crosses over, you can see that the axons come quite anterior towards the abdomen. And we call it, therefore, the anterior spinal thalamic tract when those axons go up. This carries the nocioceptive information. The axons enter the spinal cord and cross over. Then they ascend and they join other axons and form the medial lemniscus. And together they both synapse in the ventrolateral nucleus of the thalamus and then on to the somatosensory cortex. So these two separate pathways convey the information separately. And so if you get damage to one side of the spinal cord, you can actually have a loss of touch on one side of the leg and loss of temperature detection on the other leg, because the loss of proprioception occurs unilaterally to where the damage is, and the loss of nociception occurs contralaterally to where the damage is. If you cut here, then you're going to cut the if you cut on one side of the spinal cord, say the left side, you're going to get the loss of pain and temperature on the opposite side and a loss of fine control on the same side. We've talked a little bit about reflexes when we talked about the remaining function of the spinal cord after spinal cord injury. We can talk about reflexes as the number of synapses they have. So a monosynaptic reflex only has one synapse between the incoming sensory information and the outgoing movement. And the knee-jerk reflex, where your doctor hits your patella tendon with a small rubber hammer, is a good example of that, where one incoming sensory neuron synapses on one motor neuron and creates the reflex. Some other reflexes have multiple interneurons in the middle. But here's an example of that patellar reflex. When you hit the tendon, it stretches the quadricep muscle slightly, and that muscle stretch gets detected by the Golgi tendon organ, and that incoming detected stretch synapses onto a motor neuron and makes it counteract the stretch by contracting to reduce the stretch. And when that contraction happens, the leg extends out, creating the reflex. So, and when the contraction happens, the tendon is no longer stretched because it only happened briefly from the impact of the hammer. The time it takes between when the hammer gets hit and the leg goes out is a measure of the time that this pathway takes to go through the spine. This brings us to our discussion of pain as one of the important features that the somatosensory system encodes for. Pain is a very important fact of our life and important and relevant impact to our healthcare system. 30% of visits to physicians are for pain. 50% of emergency room visits are for pain symptoms. And pain has many different causes. As much as we'd like to avoid it, it's necessary, and an occasional person who's born without pain receptors experiences a great degree of discomfort and danger in their life because they get deformities, because they don't have to adjust their posture when they're sitting down for a long time. They get lots of injuries because they don't pull their hands off hot or dangerous things, and they don't have a long life expectancy. We can perceive pain in a part of the body that's no longer obviously injured, and we call this central pain. So it's pain that's not necessarily physiological in origin. There seem to be as many as 8 different kinds of pain fibers, axons carrying pain signals up to our brain. And we can even feel some of these stimuli in the absence of any known stimulus, like when we feel itches randomly throughout the day. So we talked about the anterior spinal thalamic tract being the main pathway for pain to the brain. But there's additional information that pain transmits into the reticular formation, which is associated with our arousal. pain gets sent to the amygdala, which is associated with our emotional response to the pain, and these pain signals get sent also to the hypothalamus, which affects our hormonal and cardiovascular responses to pain. And you can't just treat pain by severing this anterior spinal thalamic tract. There's remaining pain sensations even without that path intact. One very popular theory of pain and pain management is called the gate theory of pain. And it's that there's these two pathways, the fast-acting touch pathway and the slow-acting pain pathway, and that they can interact. And that the fast-acting touch pathway can actually influence how much activity gets through the slow-down pain pathway. And so it can affect how much pain is perceived for a given injury. And so when we get injured and we reach down and rub our leg where we got injured, or squeeze our finger when we just got a cut, that haptic pressure can actually reduce the perception of pain. And without any pressure, without any haptic information, the influence of pain can be increased. The way this is proposed to occur is by these small unmyelinated pain pathways that go up to the brain being modulated by an interneuron that can inhibit them. And the interneuron gets activated by the opposing pathway, the pressure pathway. So the more we rub our arm in the area where we're hurt, the more we activate this interneuron and inhibit the pain pathway. This interneuron uses endogenous opioids as their neurotransmitters, which explains the amazing pain treatment effects of opioids, because they activate this interneuron and inhibit this pain pathway. But you can get similar effects using things like acupuncture or hot showers or rubbing by using this large myelinated pressure pathway to inhibit the pain pathway. So when you stub your toe, you feel pain because the pain pathway is open. Rubbing your toe can inhibit that pathway, reducing the flow of information and relieving the pain sensation. And all these different things like massage and hot water baths that relieve pain might do so through this gate technique. The interneuron, as I mentioned, uses an endogenous opiate as its inhibitory neurotransmitter. So opiates like fentanyl or morphine relieve pain by mimicking the actions of these opioids on these receptors and decreasing activity of the pain pathways. Electrical stimulation of the brainstem in a number of places can also reduce pain, probably by closing these same gates. And our perception of pain can also be lessened from descending information from our forebrain, for instance. If we know that it's a life or death situation, the perception of pain can be lowered based off that top-down information. You'll recall in the midbrain, there's this section just anterior to the superior colliculus where the paraaqueductal gray area is surrounding the cerebral aqueduct. And this paraaqueductal gray area seems to also suppress pain. So if you electrically stimulate it with an electrode, you can use that as a pain relief during surgery, for instance. And these neurons produce their effect by exciting the pathways that project to those inhibitory neurons in the pain pathways. Oh, we'll also see how THC, that's the main psychoactive ingredient in cannabis, can also bind to receptors where there's normally endogenous cannabinoids can bind to, and has pain relieving qualities similarly by activating these same pathways that opioids do, but their effects can be synergistic. Referred pain is the idea that certain ones of our organs, while they have pain receptors, they don't have dedicated axons entering our spinal cord, bringing information to our brain specifically for the heart or the stomach. They kind of hitch a ride on other axons that send information up to our brain. And so pain in the heart is referred by our brains to the area of the arm to which those axons from the heart synapse en route to the brain. And we have a number of patterns of referred pain, like kidney pain, we feel as lower back pain because of the innervation of the axons from the kidney. The vestibular system is an important component of our somatosensory system as well. We talked about it briefly when we talked about the hearing system last week. The somatosensory system comprises two types of technologies, you could say. One are these semicircular canals, which act like gyroscopes, and the other are these utricle and saccule, the otolith organs, which act like accelerometers. So the gyroscopes detect the location of the head and ears with respect to the three dimensions, left, right, up, down, and forward and back. Based off the orientation of these three loops, fluid flows through these tunnels, moving the hairs of these inner hair cells back and forth as we move our head to the left or right. Alternatively, these utricles and saccules detect accelerations of the head, like when you're on a ride or accelerating quickly in a car. The fluid inside the utricles and saccules will push back against the back of the wall, just like you'll push back against your seat in the car, and those will be detected by detectors and transmitted to the brain. So the three semicircular canals and the otolith organs comprise this vestibular system. They tell the position of the body with respect to gravity and also changes in the direction and the speed of head movements. As we said, fluid, the endolymph inside the semicircular canals, moves as the head moves, and that fluid pushes the hair cells, the hairs, and that opens up gates and lets the action potentials increase or decrease. And the direction that they bend influences whether they'll hyperpolarize or depolarize the cell, allowing the vestibular system to code for left versus right movement of the head, for example. The utricle and saccule have hair cells also, and they have this gelatin-like substance with small crystals. So when the head's tilted or acceleration pushes it in one direction, that gelatin and crystals push against hair cells and alter their rate of action potentials. Vertigo is a disorder that the word vertigo is Latin for spinning. It's a sensation of spinning when you're not even moving, and it's associated with the dysfunction of the inner ear. And alcohol can lead to this spinning sensation, as many of you have felt when you lay down in bed at night after a couple drinks. And you can also get these similar vertigo effects just from looking off of a high cliff or up at a high building in some cases. Okay, finally, we're going to complete this lecture talking about the somatosensory cortex itself. The primary somatosensory cortex receives information from the thalamus, from the ventrolateral thalamus. It's comprised of Brodmann's areas 3, 1, and 2, and it starts to construct perceptions of what we're feeling from the incoming somatosensory information. Posterior to that, behind the primary somatosensory cortex, are Brodmann's areas 5 and 7, called the secondary somatosensory cortex. And these refined our perceptions of what we're feeling and project to the frontal cortex to make movements. So areas 3, 1, and 2 receive information from the body. They transmit that information to areas 5 and then 7. And then onwards, information goes to move the body to the motor cortex. When Wilbur Penfield was stimulating the human brain to his patients, he also observed that certain stimulation was felt as tickles or sensations on parts of the body. He made a somatosensory homunculus as well, showing that there was a map of the human skin in the primary somatosensory area in the human brain. Recently, these primary somatosensory area has been further subdivided into 4 separate areas, 3A, 3B, 1, and 2, which seem to process different aspects, different features of those incoming somatosensory signals. The slow changes, the fast changes, and at lowest level, individual muscles moving. So the flow of information, just like in the visual hierarchy, goes from the simple muscle changes to changes in skin and joints. And so information is passed from 1 on to from 3A to 3B to 1 to 2 as it gets refined and comes to represent aspects of what is being felt. If you get damage to the primary somatosensory cortex, you can have a decreased somatosensory threshold. You won't feel something tickling you. You might be unable to detect things with your hand or tell what they are. You might be unable to detect where your joints are in 3 dimensions, and you might have trouble reaching out and grabbing things. With the motor, as with the motor cortex, if you get damage, it can be reorganized. So if you get damage to the arm section of your somatosensory cortex, the area that was once dedicated to your arm can come to represent adjacent areas like the face. And this is one proposal of how things like phantom pain can happen. So here, the area of the somatosensory cortex representing the arm and the face are shown. And when the arm was damaged, the face area slowly took over that section of the cortex, creating an enlarged representation in the homunculus of that face, and a much smaller representation of the damaged arm. The somatosensory cortex also has a role in confirming that a movement has actually happened. So when you get damage, it doesn't disrupt your plans for making movements, but it disrupts how you perform them and whether you finish them or whether they get executed completely. Apraxia is an inability to complete a plan accurately or to make a voluntary movement accurately. So you are unable to reach out and grab something in a reliable way. We've talked about the visual system being divided into the dorsal and ventral streams, and this somatosensory system contributes separately to those dorsal and ventral streams. The dorsal visual stream projects, as we've seen, directly into the secondary somatosensory areas. And so the visual information about where things are and what they look like is integrated with our somatosensory representation of where our body is to allow us to reach out and grab things we need to grab appropriately. The somatosensory cortex also interacts with the ventral scheme by, for instance, when you reach out in the dark and grab something on the side of your bed, you can identify it just from that touch, indicating connections between the ventral stream and the somatosensory information as well. So here we can see that diagrammatically, the visual cortex sends information to the dorsal and ventral streams. This incoming somatosensory information contributes to that dorsal stream in order to produce movement, but it also sends information to the ventral stream to help us identify things using our proprioception. That brings us to a close for this discussion of our somatosensory and motor systems, and also for the material for this second midterm. Good luck with your study.